In order for cells to divide, their DNA must be replicated so that two daughter cells with identical DNA are produced. DNA replication is said to be semi-conservative because each daughter double helix has an old strand from the parental double helix. The first protein that comes into play is DNA helicase. Helicase unzips the hydrogen bonds that binds the two strands to each other. When this happens, two helicases activate at once, creating two replication forks expanding away from each other. This allows the other proteins involved in the process to access the nitrogenous bases. Single-stranded binding proteins, or SSBs for short, attach to each base pair to spread the two strands that make up the double helix. By separating the DNA strands apart, this allows for primase, ligase, and other proteins and enzymes to work on the sugars. An important enzyme in DNA replication is topoisomerase. Topoisomerase relaxes the tension in the DNA strand when it unzips by binding to the ends of the unzipped section of DNA. It prevents the DNA double helix from getting too tightly wound up as the DNA is opened. Primase is another important feature in the process of DNA replication. Primase puts down primer made of RNA and builds from the 5' carbon to the 3' carbon, but is read from 3' to 5'. The protein that does the actual replication is called DNA polymerase. This protein reads from 3' to 5' and subsequently builds in the anti-parallel direction, 5' to 3'. This particularity of the protein causes there to be a leading strand and a lagging strand. The strand which continually lays down primer and is replaced by DNA polymerase is called the leading strand. This strand is exposed so that synthesis is easier and replication is continuous. The other strand is known as the lagging strand. The lagging strand must continue to build from 5' to 3' on an anti-parallel strand and requires periodic starts and stops to polymerize the strand. Okazaki fragments were discovered by Reiji Okazaki and Tsuneko Okazaki. The replication of the lagging strand is made up of these fragments and are linked together by ligase. Ligase is the glue of DNA replication as it binds the Okazaki fragments together, connecting the segments of the final strand of DNA and resulting in two double helix strands identical to each other.